Hi, everyone. So in today's lecture, we are going to be continuing our discussion on plant growth and development. And we're going to be looking at how temperature affects both those processes, right? And remember, plant growth is an increase in plant size due to cell division or enlargement. And plant development is the progress of that plant through its life cycle, right? From seed to embryo to juvenile plant, mature plant, etc. But first, we are going to review. So last lecture, we looked at plant growth development and light again. And we looked at three responses that our plants can have towards light: photomorphogenesis, phototropism, and photoperiodism. And we started with photomorphogenesis, and we said it is a change in the shape or form of a plant due to light. And this is a form of a shade avoidance response, right? Shade is a threat to our plant. We know light is so important for our plants because of that photosynthesis process, right? So if our plants are getting shaded out, it's going to affect their growth and development. It could ultimately lead to their death. And we said that plants that are grown in light look different, right? Their morphology looks different. They are shorter, they are branchier, they are darker green because they're producing more chlorophyll, more photosynthesis is occurring. Plants that are grown in shade tend to be taller, less branched, and lighter green. They're not really producing chlorophyll as much. And what is regulating these changes, these morphological changes, is this phytochrome pigment system, right? And that phytochrome pigment system is able to detect red light and far red light. There are two forms of phytochrome pigment, phytochrome red and phytochrome far red, and they're able to turn into each other, right? So when phytochrome red detects red light, it turns into phytochrome far red. When phytochrome far red detects far red light, it turns into phytochrome red. And that also happens in darkness as well. And why this is important is because there is a different amount of red light to far red light depending on whether you are in full sun or if you are in shade. So in full sun, there is more red light than far red light. And the opposite is true in the shade. There's more far red light than red light. So what's occurring, why we're seeing these differences, is that in light, right, in full sun, the more red light is shining down on our plants, so more phytochrome red is being turned into phytochrome far red. And that's important because this phytochrome far red is the active form of this pigment. And that will travel to the nucleus of our cells, and it interacts with transcription factors, again, those proteins that turn genes on and off. And those transcription factors will turn on genes that will make our plant grow differently, and it'll make them grow shorter, branchier, and darker green, right? What we expect to see in our plants that are grown in light. And then the opposite is true if our plants are in shade, right? If they're getting more far red light, more of that phytochrome far red is going to turn into phytochrome red. That's the inactive version, and it's not going to travel to our nucleus. It's not going to interact with those transcription factors, and it's not going to turn on those genes. So the default growth will be this long, thin, less branched growth. So that's photomorphogenesis. Second, we looked at phototropism, which is the movement of a plant in response to light. Right now, this is regulated by phototropin, which is a blue light receptor. So it's detecting blue light. And what's happening here is that phototropin is detecting blue light on whatever angle or from whatever direction that light is occurring. And that releases auxin onto the shaded side of the plant. And auxin is a plant hormone, and one of the things that it does is that it elongates cells. So on the shaded side of the plant, there's a buildup of this auxin, and it causes those cells to grow bigger. And as they grow bigger, it tilts the plant towards the direction of light, right? Most plants have this positive phototropic response, so they're going to bend towards the light. Some plant roots will have a negative phototropic response, where they'll bend away from light. Now, we also talked about a particular type of phototropism, heliotropic movement, which is where the plant will follow, either it'll change its leaves or flower or stem to follow the movement of the sun as the sun travels across the sky. And we said that's related to the angle of incidence or the light intensity. But one of the things that it does, and one of the examples I said was our sunflowers, right? And the sunflower will actually follow the sun. What that's doing is it's ensuring that the most light energy is hitting the flower at any given time, which light is also creates heat. And that is important because bees are attracted to warmer flowers. So they're increasing their pollination and hopefully they're improving their reproduction by tracking the sun and making their flowers warmer. And the last response we looked at was photoperiodism, which is a morphological response, a development response to variations in day length. And we said we used the example of flower induction because that's pretty common when we're talking about photoperiodism. And we said there are short day plants that will flower when day length is shorter than a critical day length. There are long day plants that will flower when day length is longer than some critical day length. And there are day neutral plants that don't respond to anything or don't respond to this. And that critical day length varies by plants, right? It's specific to each species and even specific to subspecies or varieties. And it's related to the summer solstice, right? The longest day of the year. So your long day plants are going to flower before that summer solstice, right? As your days are getting longer, it's going to pass some critical day length and that'll trigger your plants to flower. Your short day plants are going to flower after that summer solstice, right? So now the days are getting shorter, and at a certain point, it's going to go past some critical day length, and it'll trigger your plants to flower. So that is it with our review. Let's get back to our lecture now. So if we are talking about temperature, what we're going to find out is that temperature will affect plants throughout their life cycle, and they'll affect those different developmental stages in different ways. And if we are starting with germination, what we see is that temperature is one of the most important factors regulating germination, right? And we've talked about germination. It's the sprouting of an embryo or a new plant from a seed. And where temperature plays a role, or one of the ways that it plays a role, is that it affects germination percentage and speed. By germination percentage, we mean the amount of seeds that actually germinate, right? So if we had 100 seeds and 90 of them germinated, our germination percentage would be 90%. And when we talk about germination speed, we are talking about the amount of time that it takes for those seeds to germinate. And we saw in our experiment when we looked at the pocket seed lab, right, that when we pre-soaked some of our seeds, they germinated faster than the non-pre-soaked, right? It took less time for them to germinate. So that is germination speed. And what we see is that if we look at a range of species, of different plant species, they have what is referred to as a range of tolerance. So that would be this entire area, this temperature range in which they will germinate, right? So your beets will germinate from about 40 degrees Fahrenheit all the way up to about 95 degrees Fahrenheit, right? They will germinate throughout this range of temperature. But they often have, or all plants have, an optimum temperature for germination, right? Where they will germinate the fastest and most of the seeds or more of the seeds will germinate as opposed to other temperatures, right? So ideally, if we're going to germinate our seeds, we'd like to keep it at this optimum range, right? This doesn't always happen in practice, but this will ensure that we have the optimum germination percentage and speed, right? When we're at that right temperature. But the further we go away from this optimum, the lower we get in these two categories, right? The lower we get in germination speed, or the more time it may take, or the less or the fewer number of seeds may germinate. So you may have a reduction in germination percentage, right? It's lower. Those two things are lower at the ends of this range. Another way that temperature can affect germination is by thermoinhibition, right? And that is if we go beyond this range, right? So if we're talking at our beets and we go past 95 degrees or lower than 40 degrees, then we will have, it will actually inhibit germination, right? So it won't occur if it is too cold or if it is too warm. And this, as you see in our figure here, is that this range is going to vary by species, right? Some like it a little bit warmer, some like it colder, etc., or they can tolerate warmer or colder temperatures. Now, temperature is also important when we are talking about seed dormancy, in particular cold temperature. And we've talked about dormancy before. It's that condition in which your seeds will not germinate under environmental conditions that are normally favorable for germination. Right? When we talked about germination, we said that we need water and the right temperature, and we need oxygen. And if we have those things, our seed will germinate. Well, dormancy creates the condition that even if we have those things, your seed is still not going to germinate. And there are some evolutionary advantages to why dormancy evolved, and we've talked a little bit about them before. But what we've also talked about, and what I want to reiterate in this lecture, is that cold or cold temperatures can be a way to alleviate that dormancy condition. And that is particularly true if your seeds respond to or have endogenous dormancy. Right? We've mentioned that endogenous dormancy is a factor in the embryo that is keeping that seed dormant. And that is different from exogenous dormancy, which is a factor outside the embryo. Oftentimes, or a common one, is a hard seed coat. Right? If that seed coat is hard and impenetrable to water, that seed won't be able to germinate, right? Because that imbibition of water, the taking in of water, is the first step in that germination process. And if it can't do that, then it's not going to germinate. So that hard seed coat is a factor outside of the embryo or the young plant that is preventing germination. And that is different from that endogenous dormancy, which is related to likely related to different hormones that are in play and maybe some genetic factors as well. And if we have that endogenous germination, one way to alleviate that is to expose your seeds to cold temperatures or a period of cold temperature. And that actual process is referred to as stratification. And let me see if I can. Stratification is this process where we expose our seeds to a period of low temperatures, keeping them moist, right? It's often referred to as moist chilling stratification. And that is going to alleviate the dormancy. And the period of time that we keep them in this, exposed to these cold temperatures, is usually several weeks, is going to vary by the species. Now, early propagators layered seeds in between moist sand or soil in boxes and they left them outside, which is where this term comes from, right? They made stratified layers of sand and seeds. And if we're talking about stratification, temperature is the single most important factor. It needs to be cold enough to alleviate this dormancy condition. So we need to keep our temperatures between 33 degrees Fahrenheit and 45 degrees Fahrenheit. We don't want them freezing because that will damage our seeds. We also need to keep our seeds moist, right? It needs to be about 20% or even 30% in terms of moisture to ensure that the stratification is occurring properly. And it's why when we did our lab, right, the method that we utilize is we're utilizing refrigeration, right? We have some modern methods or some modern technology. And it's why when we put our seeds in that paper towel, we moisten that paper towel, right, to keep that moisture in and allow this process to occur. Now, like I talked about, this is endogenous dormancy. And I said that it's the amount of time that you need to keep your seed stratified is going to vary based on the species. And we have three types or three categories that can fall into, right? There are seeds that respond to non-deep physiological dormancy, right? So these are seeds that only need a short period of stratification, only 1 to 8 weeks, or they may not even need stratification at all. A lot of our cultivated cereals and grasses and vegetables and flower crops fall into this category, right? They either don't need stratification or they only need a little bit of it. Now, a lot of our temperate region trees and shrubs, what you would find in this area, right? Your wild plants respond to intermediate physiological dormancy. They're going to require about 2 to 4 months of cold stratification. Again, in our lab that we did when I had to stratify some of our seeds, you notice these were all native plants that I chose, and one of them, I believe it was the arrowhead leaf, only requires a week of stratification, but most of them required a little bit more, right? 8 weeks or more. So we're starting to get into this intermediate dormancy. Now, some plants require even more time spent under stratification. And these are plants that have deep physiological dormancy. And examples of these are cherries or plums or peaches. So that gave us an overview of temperature and how it affects our germination, right? It's going to regulate germination speed and percentage. And we want to get it, the closer we get to that optimum germination temperature for our different plants, the better those values are going to be. And they're often towards the hotter end of that temperature range, if you guys recall from that figure. Temperature is also important in terms of germination for breaking seed dormancy, for a particular type of seed dormancy, that endogenous physiological dormancy. Now temperature, if we're moving past germination now, we're talking about vegetative growth. Just like how there's an optimum temperature for germination, there is an optimum temperature for vegetative growth and flowering in our plants. Most of our temperate region plants grow between this range of 39 degrees Fahrenheit and 122 degrees Fahrenheit. And if we go outside of this temperature range, just like if we went outside of that range for germination, we are going to have damage or death to our plants, right? If you're getting too cold, as we're going to see a little bit later, you can have frost damage. It'll destroy your tissues of your plants. If you go too hot, you can have sun scald or solarization. Essentially, your plants are getting sunburned. Too much heat can damage your cells and destroy your cells as well. So we have an optimum temperature range for vegetative growth and flowering for our plants. And really what we find is that plants will grow and develop faster as the temperature increases up to a point, right? And that point is around 86 degrees Fahrenheit. So if you are growing your plants and if you are growing them at 70 degrees Fahrenheit, if you increase the temperature, right? Maybe you are in a controlled environment and you increase it to 80 degrees, those plants are going to grow and develop faster. And if you increase it to 86, they're going to grow and develop faster. If you get beyond that, you're not really going to see any changes in growth and development. And the reason this is occurring, the reason this happens, is because temperature regulates metabolic processes, right? It regulates metabolism, and importantly, photosynthesis. And because we talked about light and the importance of photosynthesis in terms of plant growth, right? If the faster or the warmer it is, the faster and the more readily photosynthesis occurs, right? So that is how we see this change or this effect that temperature has on vegetative growth, right? It's speeding up these metabolic processes that are speeding up the growth of our plants. And if you see in the figure that we have down here, these are petunias that were planted, and they were planting them for a flowering to occur by these dates here, right? April 1st and May 15th. And depending on the temperature that they grew these petunias at, they had to start when they planted them along different dates, right? So if they were trying for an April 1st harvest, we'll say, and they were growing their plants at 50 degrees Fahrenheit, they needed to start their plants and seed their plants by January 29th. And you can compare that to if they grew them at 73 degrees Fahrenheit, they could wait all the way to March 6th to start their plants, right? The speed it takes for them to grow and develop is much faster as you increase this temperature range, right? It took about 62 days growing at 58 degrees Fahrenheit, but only 26 days growing at 73 degrees Fahrenheit, right? So really important point to remember, and a really important effect that temperature has on vegetative growth is that it speeds up growth and development up to a point, right? Up to about 86 degrees Fahrenheit. Beyond that, you don't see those increases. Another really interesting effect that temperature has on growth and development is this process referred to as diff. And this is shorthand for just the temperature difference between day and night. And how you would calculate this is you would subtract the night temperature. So say you're growing your plants in your greenhouse or really anywhere, right? And you know you're keeping track of the temperature. You can subtract the night temperature from the day temperature. And that will give you a value. Now, a low value, or if you get a negative value, right? So if you're having warmer temperatures in the night than the day, reduces stem internode growth. And what that means is that it makes your plants shorter, right? So this is a way that we can control the height of our plant. We've looked at now two different options, right? We've said that if we have more blue light, that's one way that we can make our plants shorter. Here's another way, right? By controlling temperature differences between day and night. And you can see the examples here, right? These are grown at different diff values. And again, this is just subtracting night temperature from day temperature, right? So the warmer your night temperatures or colder your day temperatures, right? The lower this value or even a negative value is going to lead to smaller plants that you see here, right? Here is a 0 value. Here's a negative value. You can see how short it is. Here's a positive value, so you can see it's much taller. And then there's another process, which is referred to as drop. And this is a refinement on this diff, right? And what they found is that you don't need to have this difference carry out throughout the entire day, right? And what you can do is you can have a morning drop, so you can lower your air temperature around your plants by around 5 to 15 degrees Celsius for about 2 to 3 hours in the morning. And that would give you a similar effect as if you were doing this entire diff, growing it at that temperature the entire time, right? So this is an example. This is besides warming your nighttime temperatures, you can have colder daytime temperatures to give a similar effect. And you don't have to keep those temperatures cold the entire time. You can just do this short morning drop, and that will result in similar effects that you see with that diff there, right? So another important way that temperature can affect our plants, or at least our shape of our plants here. And another way that we've now learned that we can control plant height using these environmental variables. Now we're going to move on to development, right? We looked at germination, we looked at vegetative growth. Now let's see how temperature affects plant development. Well, one of the big ways is that temperature is responsible in some plants for flower induction and initiation. So flower induction and initiation is the process that when a plant is mature, and we'll see what that means when a plant is mature in a couple lectures from now, right? But basically it means that when a plant becomes mature, it's able to produce flowers and sexually reproduce. So when a plant is mature, a change can occur in its apical meristems, right? The tips of our roots, or in this case, the tips of our shoots, right? Those growing regions of meristematic tissue. A change will occur in them that will switch them from being vegetative to being reproductive, right? So switch from producing stem tissue to starting to produce flowers. Now, most of our agricultural plants are self-inducive, meaning that they will initiate or form flowers after they reach a certain morphological maturity. Now, this is different, right, from our photoperiodism that we learned in our last lecture, right? In that case, it was the change in day length that was leading to flower initiation or induction. For most of our agricultural plants, they are self-inducive, so they would be those day-neutral plants, right? They wouldn't respond to changes in day length. Instead, what these plants are doing is they are reaching a certain metabolic process at a certain stage, and that's triggering them to start forming flowers. Now, people often use a calendar date to try and predict when that's going to happen, right? If you looked at the back of seed packets or information online, right, whenever you're planting anything, it'll say this many days to maturity and this many days to fruiting, etc., right? But in reality, if we want to be real accurate, research has shown that measuring the heat accumulated over time provides a more accurate estimate than counting calendar days. And this goes back to that process when we talked about vegetative growth and the effect of temperature. And that is because temperature regulates our metabolic processes, right? So if we're recording temperature, we are recording the speed as a proxy, the speed at which all the metabolism is occurring, and the speed at which our plants are growing and developing, right? And what we'll look at when we actually start looking at plant development and reproductive growth and development, we will see that we have developed something referred to as growing degree days, which is a measure of heat that plant has accumulated over the course of the day, and we can utilize that to better track where our plants are in their developmental cycle, right? Now, besides looking at temperature in terms of flower induction, and that's kind of more involved with temperature and looking at heat, right? Because the hotter it is, or the faster our plants are going to grow and develop, right? We can keep a track of that. But cold temperatures are also important for plant development, at least in certain plants, right? There's a process called vernalization, in which flowering is induced by cold temperatures. So that is a lot of our biennial plants, which have a lifespan of 2 years or 2 growing seasons. seasons, right? So they have their initial growing season where they form that basal rosette of leaves often, and they will grow for a season. They will store energy in their root system, and then they will go through winter, and the following growing season, they will grow again and they will become reproductive, and they will produce flowers and reproduce in that method. Well, for many of these plants, they require that period of cold temperatures, that winter, to occur in order for them to develop flowers the subsequent growing season. And that process is referred to as vernalization. Temperature is also responsible for dormancy breaking of bud dormancy in our trees and our shrubs, right? And the amount or length of time needed for that bud dormancy to be lost is referred to as the chilling requirement, and that's going to vary among plants, right? But our plants, as they are going through winter, our perennials, our trees and shrubs, right, they're going to require this period of cold temperature to break this bud dormancy, right, as spring is approaching. Also, temperatures and cold temperatures are utilized by our herbaceous perennials, and they undergo a number of physiological changes to survive in the winter, and those are triggered by these low non-freezing temperatures that they're going to experience in autumn, right? So our herbaceous perennials are going to go through these physiological changes that allow them to adapt to or be adapted to the cold that they're going to experience and the freezing temperatures that they're going to experience through winter. And the initial low non-freezing temperatures that they experience in autumn triggers these physiological changes to occur. So far we have looked at how temperature is affecting germination and vegetative growth and plant development. We've kind of kept it to the most part about the positive effects, essentially, right? Or showing the good aspects, right? It's speeding up germination. It's allowing for dormancy to be broken, right? To allow for germination to occur. It's speeding up vegetative growth as you get warmer. But there are instances where our temperature goes outside of that optimum range that we were talking about, or even that range of tolerance, where either they're too hot or too cold. And when that occurs, it can lead or it can have detrimental effects on our plants. So if we're talking about temperatures that are too hot, high temperatures will destroy the cell protoplasm, which is essentially everything but that cell wall, right? It's the cytoplasm, nucleus, all those organelles that we've learned about. And if you destroy that, you're destroying the cells and the tissues of your plant. You can also ultimately, it can lead to your plant dying, right? Through these high temperatures. Leaves can become solarized or sunburned when exposed to high light intensities, right? And we've talked about light and what light is. It's that energy that moves through space in waves. It's part of that electromagnetic radiation spectrum. It's the wavelength that determines the color of light. And we said that light is so important because it's required for photosynthesis, that process where plants are taking carbon in the form of carbon dioxide in the air and fixing it or turning it into sugar that it can utilize to give itself energy or to create these other products. But that light, right, that light energy gets converted to heat. That energy from the sun, all that light energy also causes heat. And if there's too much light energy and too much heat, we can have this instance where we start to see cell death and tissue death. And this is not only a problem with leaves in the form of that leaves being solarized, but we can see that in the stem tissue or the cambium layer, especially in young trees, right? They can be prone to sun scald, which that excess heat or that heat these plants are being exposed to are leading to death of that cambium layer, which is that important layer that is producing new secondary xylem and secondary phloem, the vascular tissues. So a way to alleviate that is whitewashing bark, where they put a mixture of white latex paint and water and paint it on our trees, and that can help prevent sun scald. So this actually, that sun scald, can be common in early spring that has warm days and cold nights. Right, so those warm spring days, they heat the trunks and they make the cells more active in those trunks. However, the soil is still cold and the roots are still dormant, so it's not taking, the plant isn't taking in enough water to cool off the plant. Right, one of the important things of water, it's not only utilized for different plant processes, but it helps cool the plant off. So in early spring, when you're getting these warm days, those cells are heating up and not enough water is coming off to cool the plant. Alternatively, we can have temperatures that are too cold. And when this happens, we can have frost damage. And you've probably seen this before, right? Where your leaves or tissues turn black like that. Typically occurs after the night of the first killing frost in the fall. And what is actually happening is that ice crystals are forming within the plant cells and rupturing their cell membranes, leading to cell death. And we see this by those blackened leaves that you see there. So there are several methods to prevent frost damage. Probably the most common or easiest one is to just cover your plants with either plastic sheeting or mulch or straw. And that's going to prevent heat loss, particularly at night, right, when your temperatures are colder and your plants are most susceptible to frost damage. You can also burn smudge pots to create heat that can radiate towards your plants. Spraying your plants with water, right, particularly sometimes you might have heard this or read about this at different orchards. So when water turns to ice, it actually releases a little bit of heat. And then the covering of that ice serves as an insulator to protect your plant. But you have to be careful. If you apply too little water, then there won't be enough heat that is released. But if you apply too much, then your ice can be too heavy and it can actually break or damage your plants as well. There's also a meteorological phenomenon referred to as a temperature inversion, in which normally you have warm air at the Earth's surface, and as you get higher into the atmosphere, that air gets colder. Well, in an inversion, it's an opposite. There's a layer of warm air on top that traps a colder layer of air below it, closer to the surface of the Earth. And that can, again, right, we're trying to, if it's too cold, that can damage our plants. So in that case, if this temperature inversion is going to occur, you know that's going to occur, you can utilize wind machines, where it actually helps circulate warmer air and creating warmer conditions for your plants. And the last thing we're going to look at is chilling injury. So this can occur with some of our tropical plants where they can be damaged even when temperatures are above freezing, right? So it's not as cold where frost is forming or water is freezing, but it's cold enough for these tropical plants to damage their tissues. And this can occur in a number of our plants that we utilize, more of our tropical plants, like I talked about, avocados, bananas, mangoes, tomatoes. They're all susceptible to this chilling injury. So that is it. I will see you guys on the next lecture.